In the mid-1800s, Austrian monk Gregor Mendel formulated some of the underlying principles of genetics through his observations of pea plants. Mendel noted that the plants were either tall or short. He created purebred strains by breeding tall plants together and short plants together. Mendel then crossed a tall plant with a short plant. Surprisingly, all of the offspring were tall. Mendel then tried crossing 2 of these tall offspring. The offspring from this crossing turned out to be in a ratio of 3 tall plants to 1 short plant. By looking at the underlying genetics, we can understand what Mendel observed. Information about traits is carried on genes. We can represent the gene for height with the letter T. Each plant has 2 genes for height, 1 from each parent. Different forms of the same gene are called alleles. The big T represents the allele for tall, and the little t represents the allele for short. In purebred plants, both alleles carry the same information, and the plants are said to be homozygous. When tall purebred plants are crossed with short purebred plants, their offspring each receive 1 allele for tall and 1 for short. The offspring are called heterozygous. Since these plants all appear tall, we say that tall is the dominant trait and short is the recessive trait. When these heterozygous plants are crossed, 3/4 of the offspring carry the dominant trait and appear tall. The recessive trait appears in the remaining 1/4 of the offspring, who receive 2 alleles for short. This type of simple inheritance is one of the many ways that traits are passed from parents to their offspring.